Functional genomics and proteomics. In molecular biology, this activity, it mainly deals to study the functions of different genes that are expressed from one tissue to another tissue or within a single cell. So overall, we can say that it is mainly related to study the expression profile of different genes, that which gene it is expressing at a particular time period and in which tissue. So overall, it mainly deals with gene-to-gene interaction and their functions. So different techniques have been employed to study the functions of genes, and one of the most extensively studied technique that is currently in use, it is the DNA microarrays. DNA microarray is a technique that allows us to study the expression of thousands of genes simultaneously. And here, the basic protocol of Northern blotting, it is used. If you remember, we studied different blotting techniques that can be used to study for the detection of DNA molecules or RNA molecules, like the southern blotting or northern blotting. Northern blotting, it is mainly preferred for the detection of RNA molecules. जैसे कि मैंने बताया कि DNA माइक्रो एरेज का जो प्रिंसिपल है मोर और लेस वो सेम है जो कि हम प्रीवियसली यूज करते रहे हैं नॉर्दर्न ब्लॉटिंग के साथ, कि इसमें भी हमें DNA प्रोब्स चाहिए होते हैं विद नोन सीक्वेंसेस. तो इसमें जिन जीन्स का सीक्वेंस हमने मालूम करना होता है या जिनकी डिटेक्शन करनी होती है कि कौन सी जीन जो है वो किसी टिश्यू के अंदर एक्सप्रेस कर रही हैं, तो उन जीन के हमें सीक्वेंसेस नोन होने चाहिए. So that the known sequences of the genes or the number of genes, they can be immobilized on the solar surfaces. So here, nowadays we prefer to immobilize the genes or the strands of genes that may range from a few dozens to a few hundred nucleotides in length. So such gene fragments, they are immobilized on the solar surfaces with the help of robotic devices, so that very minute quantity of DNA, it is being immobilized on the surface, maybe in nanoliter quantity. And after immobilization, then the DNA that is extracted from the single cell, or in other cases from the tissue, it is extracted, and then this messenger RNA, it can be fluorescently labeled by using different type of reagents. So here the radioactive probes can be used, or we can use some fluorescent dyes or fluorophore for the labeling. So here probe may be labeled. So in this case, the DNA or messenger RNA that we have extracted, it can be used as a probe. And this labeled probe from the sample or the messenger RNA that have been expressed in the tissue, it can be used as a probe for the hybridization with the known gene on the glass slide. So that when hybridization is there, it will result in the emission of light of particular color and wavelength. So when hybridization is strong between the immobilized DNA and the probe, that is the target DNA from the sample, then this emission, it can be detected in the form of signaling. And hybridization on the glass slide will indicate that the genes, they are present in the sample. So दूसरे अल्फाज में हम ये कह सकते हैं कि अगर हाइब्रिडाइजेशन हो जाए सैंपल के मैसेंजर आरएनए से, जिसको हमने एज ए प्रोब यूज किया है विद द नोन सीक्वेंस ऑन द सोलर सर्फेसेस, जो कि हमने इमोबिलाइज किए हैं, तो इसका मतलब है कि वो जीन जो है वो टिश्यू के अंदर एक्सप्रेस कर रही है. So in this case, where we have immobilized the DNA, such glass slides, they may be given different names like the DNA chip, biochip, or other naming have also been used. Now, what is the basic protocol that we can explain diagrammatically for the explanation of DNA microarrays? So here is the comparison of two population of messenger RNA that have been extracted from the single cell and the later stages of development of the frog. So we can expect that messenger RNA population, they can express differentially in these two tissue types. So messenger RNA have been extracted, and then by using reverse transcriptase, they have been converted into cDNA. And one population of messenger RNA, it was labeled with green fluorophore, and other it is with red fluorophore. And in the next stage, after converting into cDNA, they are then immobilized on the solid surfaces, like here. For example, here different spots or circles on the glass slides, they belongs to different set of genes that have been immobilized. And when we will hybridize our sample in the form of messenger RNA from two cell tissue types or single cell, then hybridization will result in the emission of particular color. For example, if messenger RNA it is from single cell, then the green spots, they will be visible on the glass slide here, and otherwise pink or red spots will be visible in case if the messenger RNA has been hybridized from other developmental or later developmental stages here in the form of pink or red spots. And the yellow spots indicate the genes that expressed at both cellular organization, that is at single cell stage and at later stages of development. So here we can know the expression of different genes at single cell stage or at multicellular stage. Now this diagram, it indicates the expression of 6200 genes from yeast that have been exposed to different culture conditions, that is normal culture conditions, the spots that are indicated as green one, and the genes that have been expressed at later stages of sporulation, they are indicated in the form of red spots. And yellow spots, they belongs to genes that express constantly at the cellular or even at the sporulation stage. So in this way, in the yeast genome, we can detect different genes that are expressing in the normal cell cycle or at the later stages of sporulation.